Hi everyone, welcome to Foresight's Biotech and Health Extension Salon, sponsored by 100 Plus Capital. We're really excited to have Roman Bauer here today. Roman, you are a Foresight Fellow, and today you will be presenting on computational modeling and biomedicine with the X in multiple baskets. We're dying, hopefully not dying literally, but dying as a figure of speech to hear more about what that entails. So please take it away and I'll share a lot more about you in the chat. Thanks a lot for joining.
All right. Thanks, everyone. And thanks the organizers for having me. It's a pleasure to be giving this presentation about computational modeling research that I've done in the past relevant to biomedicine. And so before I start with the more specific talks, I'd like to briefly discuss the relevance of computational modeling in general for this kind of research. In computational modeling, usually the approach we take is that we have some kind of system that we are investigating or studying, and we are creating some model which can encompass a large variety of different types of models, and then we essentially run the model and check whether it agrees with the experimental data. If yes, that's great. Then we can also make certain predictions, which is one of the major objectives of such a computational model. And we can then test the model. And if it still works well, then we can do the next iteration and refine the model. Of course, if it doesn't work well already in the first step, then we have to refine and go back to the drawing board before. But so this is a general approach. And in this definition, computational modeling can also include AI. So I just wanted to avoid this kind of cliche of just directly mentioning AI. But it's a very general term and can include AI methods like deep learning and so on, or machine learning methods, but doesn't necessarily have to. It's a more general approach. So I'm particularly interested in the brain. I'm a computational neuroscientist, and the brain is obviously a very complex system. And so the question that arises is, what kind of general approaches are there to save the brain, to rescue the brain from deterioration, from processes of aging, and so on? And I see at the moment three main directions to go. One is to directly address age-related deterioration and disease of the brain, which is a naturally occurring phenomenon. And another part or another direction would be to repair and regenerate the deteriorating brain. And for other organisms like the starfish, for example, have various astounding capabilities for regeneration. And so it would be very interesting to see if we can maybe leverage similar mechanisms to regenerate the brain. And also there is biostasis, which is again quite a general topic, which can include emergency medicine-based approaches to essentially slow down metabolic processes, to then gain time to do some interventions. And ultimately, it could also encompass slowing down metabolic processes in the long term. And so it's a very, again, overall research direction. So regarding the first direction, I want to just show some of the research we have done in my lab and with collaborators. And this will then allow to essentially paint a bit of a picture, what are important topics at the moment, what are challenges, and so on. As discussed, the brain is a very complex system, and we know that it undergoes significant changes normally during aging. For example, here on the right-hand side, a measure that indicates connection strength in the brain from diffusion tensor imaging. And we can see there is a clear trend of decrease in those indicators with age. And so one paper that we published relevant to this topic is this one here, where we used data from diffusion tensor imaging in order to create a computational model of the brain undergoing changes during normal aging and also during Alzheimer's disease progression, because there are differences, of course, in the brain structure in both conditions. And we did this by using data from the ADNI database, which is an public database where researchers can gain access to high quality data. We had access to both controls and Alzheimer's disease patients. And essentially, we employed a machine learning analysis approach in order to create a predictive model if a subject would be undergoing Alzheimer's disease progression or not. And so what we did is we essentially simulated the time course of the aging brain in the normal case, in the normal aging model, and in the pathological Alzheimer's disease aging model. And the goal was to leverage this kind of progression model to differentiate as early as possible between the two cases, between the two scenarios. And we obtained quite good performance in differentiating this. And as you will see here on this figure, we have classification performance that essentially indicates the performance of classifying between the two classes, and at stage -5 approximately, which is approximately 5 years of time. So one stage refers approximately to 1 year, and we could obtain quite high performance. So these are different indicators of classification performance at around -6 or -5 years before current clinical diagnosis. So this is an estimate of how good our performance would be compared to current clinical diagnostic performance, which happens later, as we believe, based on what we could achieve by looking at the brain structure from diffusion tensor imaging. And this is the statistical significance indicated by the p-value. And again, that at around -5 years or -5 stages, we achieve very high statistical significance. And so this is quite encouraging to see that by looking non-invasively into the brain structure, because diffusion tensor imaging is an MRI technique and it is non-invasive, so we can obtain very valuable information from the brain structure across different ages, and this helps for diagnosis based on this model probably earlier than what is currently doable. Another way to look into the brain is by using EEG. So with EEG, we can measure neural activity, and again, we can computationally analyze it. And we have published also on this modality to essentially, again, classify EEG data from healthy controls versus subjects with certain types of dementia, including Alzheimer's disease and Parkinson's disease dementia, and dementia with Lewy bodies. And so what we did there is we followed a different approach, which is quite unique, because what we did is we looked at the ratio of EEG activity indicators in two different conditions, namely while the eyes are open. So people had either their eyes open or eyes closed while their activity was measured, the brain activity was measured. And we looked at the ratio between those two. And it turned out that this ratio is highly informative on whether there is a pathological case or not. And one interesting fact of this is also that this is like a natural personalized indicator, because it compares those two conditions for each subject, right? So it's in a way going to the direction of personalized medicine. And so this is more informative than just looking at one condition. And as you can see here, by looking at healthy controls, so HC stands for healthy controls, we see huge differences in those ratios as compared to Alzheimer's disease subjects, for instance, which had here much lower values for those indicators. So based on this kind of ratio indicator approach, we could classify different types of dementia very accurately. As you can see here, this table summarizes it just based on EEG analysis, which is, again, non-invasive. This is one of the major benefits of EEG, is that it's something that is doable in a non-dangerous way. It's safe. It's something that is very practical. It's cheap. So this is something that has very promising avenues for future applications along those lines. And other people have also used EEG analysis, for instance, to look at the aging brain directly in normal aging. And as you can see here, so those colors indicate information, content, or relevance for age prediction. And as you can see here, there is a complex profile across the entire scalp. And so this is another piece of evidence that shows that, yes, indeed, there is a lot of things happening that we can objectively quantify, but we don't really have a mechanistic understanding of what exactly is going on there. And so we think that there's a lot of research that needs to be done to really get to the bottom of this, rather than just observation-based diagnosis, to really understand why are those changes the way that we see them, and what can we do about them ultimately, because this is about diagnosis. But obviously, if you want to really address those challenges of brain aging, we need to do something about this. And this is just the first step, is the diagnosis, but then later on we need to also remedy those changes. Another type of modality that we have used more recently is retinal fundus imaging. So we have looked into AI-assisted approaches and techniques to analyze the retinal fundus, which is the neural tissue at the rear of the eye. And one can image also the vessels there, the arteries and veins. And one can use AI to do computer vision, quantification, and prediction of different kinds of neurological conditions. And as shown in those papers here that we published rather recently, there is very promising information there that can allow us to get an indication of those diseases. We need to now, what we are currently doing is we are corroborating those results on larger cohorts and improving the overall performance. But this is also a very interesting direction because, again, retinal fundus imaging is very cheap. It's non-invasive. It's very practical to be done. You can even do it with portable devices and so on. And there are changes happening in the vasculature. So this was about the first direction. Now the other direction, the second direction that I listed initially, is with regards to repair and regeneration. And if we want to accomplish regenerating the brain, we need to understand how the brain develops, because the brain undergoes significant changes, and we all start from a single precursor cell. We have all of those different important stages, and if we want to really repair parts of the brain, we need to have an insight into how these parts of the brain develop naturally, because the brain is extremely complex. It has 100 billion neurons and a million billion synapses. So 10, this is a 1 with 15 zeros of synapses, of connections between neurons. And in regenerating the brain, we essentially would follow the same approach as nature does when it constructs the brain, which is that it leverages the genetic code, right? The genetic code has all the instructions implicitly encoded in it in the form of the DNA that tells each cell what it should do. And if we want to regenerate the brain, we need to leverage the same approach, which means that we need to understand how the brain develops to effectively do that. And while the brain develops, what cells do is they have genetic instructions, as I just said before. And those genetic instructions can be understood as rules that tell each cell what it should do. And those cells interact with the local external environment where we have chemicals, we have mechanical interactions, we have electrical activity, and so on. These all play a role in what the cell does and how it behaves. And so just to give a little introduction into neuroscience, just because I suspect not all here may know those fundamentals potentially. So a neuron can be simplified as a cell body or soma that has dendrites and axons, which allow electrical activity to be transmitted. And this is how you get those neural networks that are connected by synapses between those axons and dendrites. And we have used computational modeling to gain insights into how those neural networks develop in a biologically realistic manner. So, for example, here in this paper that has been published now a decade ago, it was one of my first papers, where we showed that we can simulate realistic connectivity formation in vitro, so in 2 dimensions in this case, between excitatory and inhibitory neurons. So the red neurons here would be excitatory, purple ones inhibitory neurons. And you can see here very complex structures emerging, all from very simple growth rules. And the size of the genetic code is indeed very small as compared to the complexity of the brain. And if you understand better how the cells grow their neurites, their axons and dendrites, and how they connect with one another, then this can give us insight into how this complexity develops and how one can repair it ultimately. So I just want to give one example where we have used this approach to explain the development of a brain architecture, which has been experimentally observed many times, many decades ago already. And this architecture is the so-called superficial patch system. So injections of neural tracers in the superficial layers of the cortex, one can inject, for example, horseradish peroxidase, which is a tracer that allows to label neurons that connect to the injection site. So this would be the injection site here in black, this black patch here. And you see here that those neurons here in those patches somehow connect to this injection site. So it's a very non-uniform connectivity, right? Somehow, for some reason, neurons here in the empty spaces here don't connect to this patch, but the others do. So we don't really know why exactly, but we know that the mammalian cortex has this kind of connectivity structure. And so if we would better understand how it develops, then potentially we could also understand how it could be repaired or regenerated. So it turns out that one of the clues that can explain how it develops was given by the famous computer science pioneer Alan Turing. And here I am next to Alan Turing in a park in Manchester. And what he published, and this is less known than his seminal work on the Enigma and of course the role they played in the Second World War and so on. But he published this seminal paper, The Chemical Basis of Morphogenesis, where he explains that interacting substances can form in a self-organizing manner very complex patterns and stable patterns. And another model that builds on this work, the so-called Girer-Meinhardt model, showed that you can do this with two substances, an activator substance and an inhibitor substance. And so what the activator substance does is that it is antagonistic. So the more of it is produced, the more it enhances the production. So the more there is there, the more it produces. And it, on the other hand, it also increases the production of an inhibitor substance, which is antagonistic to it. So the more of it is present, the less of the antagonist of the activator substance is produced. So this is a very simple model, but it's very powerful from an explanatory perspective. So what we did in this paper, which was actually my first paper ever published, is we simulated the activator-inhibitor model of Girer and Meinhardt with so-called agent-based modeling, which is a computational approach for simulation and modeling. And we had cells that represent neurons in the developing cortex. And those cells produced those two substances, and we could indeed create patchy patterns. So yellow means that there's a high concentration of both the activator as well as the inhibitor substance. And then the neurites projected, they were drawn towards high concentrations of this substance. And then they created those connections or branching patterns. And here you see a video of this happening in a self-organized manner, because this is like an ultimate test to show that indeed this is self-organizing. There is no global supervisor needed, but all of this can happen just based on a genetic type of encoding. And we used actually several activator-inhibitor models, which is why you see different colors. And then you see here those patchy patterns developing. And also what is that this looks quite nice aesthetically, which is why we were also on the cover of that journal. And you can see here this patchy pattern similar to what I just showed you at the beginning with those injection sites, with the injection site. Now, this was just more or less qualitatively, but then we also confirmed that if So people have shown that the patch width, so if they looked in different species and in different parts of the brain, what is the width of those patches and what is the spacing between patches? And they could see a very strong linear relationship between those two variables with a slope of approximately 1.7. And we could confirm the same slope and same linear relationship just by changing a single parameter in our model. So this is further evidence that indeed this model is very plausible, and it captures several of those quantitative indicators as well. And now this is, of course, a first step again. It shows us how it could develop, but then there are many questions that now arise if you want to use this insight in order to potentially regenerate this system. And one would be, how can we reproduce this patchy pattern of morphogens and so on? So there are still many questions that need to be addressed, but I think this is a nice first step that shows the power of computational modeling and computational simulation. And this is the cover image that was there with that publication. Finally, as I said, biostasis is also another very important and very promising direction to go, because if we are, for example, in an emergency situation where maybe there's organ failure or so on looming, then it would be a great intervention if one could slow down everything and then help the patient. And it's an open question, of course, how long this should be done. The longer the better. And we have published this review a few years ago where we summarize the state of the art and also raise challenges, and also a little bit reflect on computational directions there. So computational ways to improve cryopreservation, which is a form of biostasis. And I would like to raise that this is really something that is on the research agenda of many institutions. And so it's really something that has attracted and still attracts, of course, and it's increasing in attraction in terms of promise for future biomedical interventions. DARPA, for example, is interested in this. There has been research on pig organs and so on brains, and various other lines of research that touch on biostasis or are relevant to biostasis. So there's a lot of things going on there. Also for space travel, this would be a great technology to have. If you think, for example, about travel to Mars, which would take more than 6 months, I think at least 6 months. This is very difficult to accomplish if you have many people that need to live together in a small space. There is also a lot of applicability there. And again, I think computational modeling can play a huge role there. It has already led to advances. But I would like to highlight one of the projects that I've been working on and which we have applied to cryopreservation as well, which is the Biodynamo software collaboration. We have published this work. Biodynamo is a consortium of multiple different institutions. And I'm the spokesperson of this collaboration. And we have now, I think we are eight institutions. And we do research in computational biology. And we are very interested to engage with other stakeholders and institutions who want to conduct research in computational biology using agent-based modeling, which is this approach that I mentioned before, where we essentially model cells and cell elements as individual agents that behave in some way in a three-dimensional physical world in order to reproduce biological complex systems and to gain insight into their mechanisms, and also then to potentially improve the state of the art. And what is also important is that we can actually model neural tissue with biostasis. It is a very performing software that allows to simulate millions of neurons. We have simulated billions of elements of cells with Biodynamo. It's arguably one of the most performing, if not the most performing agent-based simulator in the world. And the nice thing is that it can also model very heterogeneous, complex neural networks and neural tissues. And computational modeling, such as with Biodynamo, is relevant for cryopreservation because cryopreservation is a very complex topic, because you need to consider a lot of variables, right? You need to consider, for example, how quickly do you cool down the tissue? What kind of cryoprotective agents do you use? What kind of concentrations do you use? How do you administer those cryoprotective agents? At what times? Because if you administer them at lower temperatures, then toxicity is reduced. So toxicity is a massive issue in cryopreservation, as you can damage cells, of course, but it's lowered at lower temperatures. On the other hand, then also diffusion is lower. So you have a balance, you have to strike a balance. And then there is also the question of how do you thaw the tissue. And so there's a lot of variables there, and computational modeling can help address this complexity, because it's a way to bring together, to integrate various types of experimental data into a coherent framework, and then we can optimize those parameters in order to improve those protocols. Because at the moment, cryopreservation is mainly an experimental discipline. So people don't usually use computational optimization or AI to improve those variables, but it's more an empirical process that requires a lot of trial and error processes. And so we argue that if we could bring in a computational component here, we could render the entire research pipeline more efficient. And indeed, we have some very promising results there. My PhD student, he just recently finished his PhD successfully. He created a computational model that predicts a better way to cool down cells in suspension, because there are different ways of how you cool down, right? You can linearly cool down, or you can first very fast and then slow, or the other way around. So there are infinitely many ways of cooling down. And he created an optimization algorithm that predicts a better way to cool down cells in suspension. In this case, these were Jurkat cells. And here you see the state-of-the-art methods that have a linear cooling rate, so 0.5 degrees per minute, or 3 degrees per minute, or 10 degrees per minute. And then this multi-step approach that we will hopefully soon publish has a much better cell count after cryopreservation. So the post-thaw viability is enhanced when using this multi-step optimized approach. And so this is just one of multiple, of course, computational results and modeling efforts that show that cryopreservation can benefit from computational modeling. Of course, and also this particular research here, we need combination of both computational and experimental research, right? We need to create the data to inform the computation models, and then afterwards to refine them. Because as I said, we need those predictions in order to be able to say that there is really something valuable in a computation model that we can then further refine later on. And so get into this iterative circle that I explained at the beginning of this talk. So to conclude, there are different approaches to use computation modeling to help with biomedical applications, to address issues of the aging brain in particular. And there is a lot of funding going into particularly the aging brain. And this is a very complex process also with regards to the development of the brain. So as I mentioned, the entire process of development is a complex process, and that needs computation modeling to really formulate hypotheses. Because at the moment, no human can understand all of those mechanisms and processes that play out during brain development. And so we need a computation model to formulate and quantify brain development in order then to also address potential treatments that help regenerate or repair the brain. And with regards to biostasis, there are many initiatives, large-scale initiatives also that address biostasis-related topics. And I think at the moment, most of the funding goes to the first direction, right? When in fact, I think there is a lot of potential also for the other ones, in particular the third one, which I think compared to the other ones receives much less funding support. And I think computational technologies can help in cryopreservation particularly, which at the moment isn't done a lot, but I think it can definitely leverage current technologies. As you all know, AI has made huge advances, and I think computational technologies can leverage those advances also for cryopreservation research. And so I would like to end with this kind of conclusion that we should not put all the eggs in one basket or all the funding in one basket or in one direction, but it makes sense to be more balanced because the other approaches also have a lot of promise, and we should definitely, I think, consider this more. With that, I would like to thank you for your attention and all the people that have been involved in this research, all the funders. I'm very grateful also to my lab that is very proactive and has kept me on my toes in my research as well. So thanks all, and I'm happy to take questions.
Fantastic. Thank you so much. That was very informative. Thank you. Okay, if people want to raise their hand and to ask a question, feel free to go for it. Another alternative is if you cannot unmute yourself, you can just pop your question into the chat and I'm going to ask it for you. And yeah, we'll just go for a few minutes of that. If you're okay to take questions now already, there's the first hand up already. Roman, is that fine?
Yes.
Yeah? Okay, cool. Then Chris, why don't you unmute and ask your first question?
Hi, Roman. Really fascinating work, and I wasn't aware of it, so I'm glad I'm attending. I've done 15 years of modeling of neurological disorders and how to treat them with electromagnetic fields. So I can relate to some of your work, but I can't wait to read more about it. Regarding, cryopreservation, I've been peeking in at that for many years, and it seems like a lot of progress has been made on the cryonic side. But if we don't know where memories, exactly how memories are stored, where they're stored, how cognitive functions are stored, encoded, there's a big gap there. I'd love to hear your comment on that.
Yeah, I agree. Thanks for this important question, which is definitely in the center of the biostasis direction. And I agree. I think there is a lot of topics that are also from a neuroscientific perspective unanswered. But I think the main challenge will be, at the moment, the main challenge is to be able to cryopreserve complex tissues, because we are still quite early on in terms of cryopreserving tissues such as, for example, like the retina. I have myself worked on the retina, which is a neural tissue. And I could see firsthand that this is already a difficult topic, even though it's a very small tissue, but it was very challenging to get some halfway looking retinas after cryopreservation. And so I think, yes, it's true, as you say, that we don't really understand how memory exactly is formed. And this is a challenge also for cryonics. But even before that, I think we need to first address the question of how can we cryopreserve complex tissues, even just the liver or the kidney, right? If we can cryopreserve those. There was recently some advances, significant advances. I think it was the rat, I think liver, but it was an organ of the rat, I think, with nanoparticles, and that was a great progress. But we need to still scale this up to larger organs, and so the question still remains: how can we, in general, improve cryopreservation before we really address cryopreservation of the brain? I think the priority should be now: can we accelerate cryopreservation research in general to study, for example, just the cell interactions? How are cell interactions affected after cryopreservation? There is a lot of things going on that are probably affected by cryopreservation in terms of crystal formation, if there are crystals. And how do you perfuse, ideally? How do you optimize perfusion? There are so many questions, even before we look into the brain, I think, that need to be first addressed.
Thank you.
Any follow-up? No? All good? Okay, wonderful. Maybe I will ask a few questions. Those are more the general questions that the Foresight audience cares a lot about. You've already given a bit of both like the history and current challenges and a bit of an outlook, but if you were thinking more ahead in terms of like, in an ideal world, if the technology progresses as well as it can be, like realistically speaking though, where do you think we could be in 5 years? What do you think is possible, both like specifically maybe on a biostasis, with a focus on biostasis?
Yeah, that's a tough question, because as predictions are very difficult, especially if it's about the future. Yeah, and I think we really need some more investments into biostasis, because at the moment things are still in a linear phase in terms of progress. And so I think what is very good to see is that there is also now more commercial interest as compared to 5 years ago, for example, into cryopreservation and cryonics particularly, right? And for example, Tomorrow Biostasis and so on is working on cryonics as well. And so I think there is now definitely more public attention on biostasis. And I couldn't make a definite projection about in 5 years, but I think if there is really some more funding, I think we can make a big step ahead, potentially even for cryopreserving larger volumes of certain types of organs. But at the moment, I don't think we are very close to whole organism biostasis, for instance. I think this is further than 5 years away.
And 10 years? What about 10 years? Or when, I guess, hard to put your finger on it, but what would make you feel more confident that we're on a good path to biostasis in the next 5 years? Maybe that's a better question to ask. What would need to change? I guess the funding is the biggest part that you pointed out, right? Yeah. Any other bits, like any scientific, technological breakthroughs that we really need in the next 5 years? Any areas that you want to pay attention, or like draw attention to?
Yeah, there is one promising direction which I'm also engaged in. I'm co-founder of a company called Oxford Cryotechnology, where we employ AI for cryopreservation research. One of the first objectives is to use AI to improve cryoprotective agents. I think there's a lot of untouched potential in terms of using AI for molecular design of CPAs. And I think what we need there is I think this is one of the low-hanging fruits, because we would need essentially just some base data set that would allow us to improve our AI predictions. And so if more people would come together, and I'm reaching out to anyone who is interested in this, it would be great if we could join forces to create such an important base data set in order to leverage AI effectively to improve cryoprotective agents. Because we still don't really entirely understand how cryoprotective agents work. How exactly are they preventing injury from cryopreservation? There's also different cryopreservation procedures. There is a slow cooling, and then there is a vitrification, and then CPAs come in different types. Some of them are penetrating, some are not penetrating. And depending on the context, the protective impact is different in its nature. And so I think there's a lot of things that could be done with AI, especially given all those vast improvements in recent years, to leverage such a data set. So I think maybe we just need improved networking and better collaboration capabilities to achieve a better cryoprotective agent implementation.
Awesome. Thanks. Regina, you had your hands up.
Hi.
Regina. Great. Yeah, yep, we can hear you.
Okay, very good. Hey, Roman, I had a quick question. I was fascinated with what you were saying about understanding or recapitulating the structural development of the brain using agent-based modeling. I've actually had an interest in that for a while, and I was wondering if you could point to some, I know that you cited some earlier papers, but maybe you could say a little more about what you've done and where you might be going with that. I think it's a fascinating area, and I love the idea that following those very simple rules, you get this enormous complexity in these self-organizing systems.
Yes, thank you, Regina. And yeah, good to have you on the call.
Yeah, thank you.
Regarding this part, this is actually my main research. My main line of research is on computational modeling of neural development. And essentially what we're doing is, as you said, is we are leveraging the fact that the brain develops from a single precursor cell, which has the instructions encoded in the DNA. And it has been estimated that the genetic code has an information content of approximately equal to about 1 gigabyte of information. I'm a bit skeptical about that, but I don't think that it's much more than that or much less than that. So maybe it's an order of magnitude more, or maybe even a bit more. But compared to the complexity of the brain, it's definitely less, right? So there's a huge difference in terms of complexity. So as a computation modeler, why not use this, right? Why not leverage this very powerful principle to simulate the brain ultimately? And agent-based modeling is, I think, the best way to do this, because it naturally incorporates the capability to leverage local information exchange. So local information exchange is what is, from a computation perspective, very attractive because it's easily parallelizable, right? If you model something complex, but you have a lot of interactions between the different parts of your system, it's very difficult to simulate it. But in agent-based modeling, you can parallelize those systems. And we have done that actually with Biodynamo. We have parallelized it, and we can use high-performance computers in order to simulate brain development. And one of my PhD students is actually currently finalizing his work where he simulates an entire column of the cortex with agent-based modeling, and he gets very good results in terms of the synaptic connectivity, in terms also in some very promising results in terms of activity, the electrical activity. And so we just need to essentially continue, I think, this line of research to scale up the entire system, and then ultimately to integrate more data. And there is a lot of data available. This is also one of the nice things. For example, the Allen Brain Project has a lot of data on brain development in terms of gene expression and morphologies and so on. Or there's the Neuromorpho database, which has neural morphology and so on. So there's a lot, everything is here. We just need to do it, right? We just need the people to do it.
I would love to talk to you more about this. And did I just hear you say that one of your students recapitulated the structure of a cortical column?
Yes.
Using modeling? That's amazing.
Yeah,
okay.
Thank you. Yeah, we should have a chat. Definitely.
Yeah. Okay. That sounds good. And I just want to also add that there's a scientist, Bill Bialik, and he has he's interested in the similar questions, but he also uses morphogens to describe why different areas of the brain have completely different structures. And I think that could add a lot to the modeling, the agent-based modeling that you're doing, because I'm assuming each agent is a single neuron, but the brain not only has these different areas, right? That's thalamus and the Broca's center, whatever, but they have different types of neurons. But if you introduce a layer of morphogens, that could help each particular section differentiate beyond the information that you would imagine was available from the simple rules that you're using to do the agent-based modeling, if that makes sense. Anyway, I find this fascinating. I don't want to take the discussion too far away from its base, but I'd love to talk about it more. So thank you.
Thank you. Yeah. No, that makes very much sense. If I just may add a piece of information relevant to that, there is the so-called protomap hypothesis from Rakic. Pasco Rakic, he has formulated this hypothesis that essentially the cortical development happens via morphogenetic specification in the early stages in the cortical plate, which is then retained when the columns develop. And we have built on this hypothesis in that paper that I referenced here in this talk from 2014. So yeah, there is a lot of conceptual progress in terms of explaining this heterogeneity that you referred to, right? These different brain areas in terms of early developmental processes, such as the protomap hypothesis. And so, yeah, definitely we can talk about how this could be taken forward. Yeah.
Awesome. Thank you.
I'm glad you joined the call. I guess as we're nearing the end here, like one thing that I really want to know from you, Roman, is what do you think, what's on your plate in the next year? What are you going to focus on really in the next year or so? And how can people in this call or other people watching on YouTube afterwards help you in your work if they're excited about what you're doing?
Yeah, my plan is to continue research along those lines. I'm a believer in the approach that we need to fight on all fronts. So I'm interested in all those three directions. I just think we need more support for the second and the third as compared to the first. I think it's currently unbalanced. And I want to just do research. That's my passion, right? And it's difficult to predict where exactly this leads me. It could be that next month there is some idea that I really want to pursue. And it's difficult. I think we have to be a bit spontaneous in research, I think. Otherwise, it's not really research anymore. It's like industrial, maybe innovation or something like that. And
no grand 5-year plans that you're going to follow to the bit.
No, not really. Yeah, I play by ear, and yeah, I don't know if it's a good thing, but this my take on things.
Is there a biostasis roadmap? I know that Rob Frieders has this book on nanotechnology and how it can benefit biostasis, but is there like a proper biostasis roadmap that's trying to unite the field around like critical milestones and some timelines and capability development?
I'm not sure. It's a good point. If there is not, I think there should be one. Yeah, I think it would. But that's maybe something we should definitely work on. Yeah, yeah. I think it sounds great. At the moment, I think it is a bit segregated the field. People have different. Some are more interested in, for example, also not necessarily cryopreservation, but fixation, for instance. Others are interested in vitrification, but not slow cooling, and others are interested in slow cooling, but not vitrification. I think there is still quite some segregation. If we could maybe come together. Actually, I'm organizing those regular biostasis update meetings. Also, Regina is part of, maybe that's a way to also get in touch and collaborate in the future. I'm just interested, if anyone has ideas or would like to collaborate or engage with this research, then feel free to get in touch. You have my email address, and otherwise, there are lots of ways to get in touch.
Awesome. Thank you so much on behalf of everyone who listened. I really appreciate it. And whenever you have an update on your work, please reach out and we'll have you back for another. So thanks, everyone. Thanks for your great questions. Have a good rest of your day.